Wonderful. So on behalf of the Single Cell Spatial Analysis Program, it's my great pleasure to welcome Raheleh Rahbari to speak with us today. She comes from the UK, so it's a little bit later there at the Wellcome Sanger Institute. She got her PhD at the University of Leicester in 2012, and since then has been at the Sanger, first as a postdoctoral fellow with Matt Hurls, and then with Thierry Voets, and now as a group leader since 2019. So a young investigator off to a great independent start now. She's interested in basically mutational mechanisms and spectra in the germline and somatic cells and cancer and normal biology over lifetime. So a lot of issues that relate to my own interests in mutation accumulation. I will mention that she's got a lot of great publications in the area. I just quickly highlight just a couple of that really to me connect to some of my reasons for being very thrilled to host her in this particular series. First was a 2016 paper in Nature Genetics with Matt Hurls using a large series of specimens. And I imagine this might be sort of at least an introductory component of what she'll tell us about today. I'm looking at, again, the spectrum of mutations in the human germline as a function of age. That was really a seminal paper that set the stage for a lot of future work. More recently, many of you will have seen in September of 2021, this year, there was a series of, I believe, 5 papers, and Raheleh was an author on 2 of them, senior author on one of them, that was really focused on using high throughput sequencing to really address issues that I think I want to be part of the single cell spatial analysis program, which are issues of cell lineage and placement of mutational cell lineages in tissues. And that really was, that series of papers was really what put us over the edge to actually invite her to speak in this series. And then last, I just wanted to quickly mention a paper, because I'm not sure she will, but a paper that she mentioned to me has recently gotten accepted into Nature, I believe, if I remember correctly, that's looking at the effect of environmental agents on germline mutation accumulations. That's been on bioRxiv and has created quite an interest in the environmental mutagenesis area. So just a few highlights of some of the reasons that we're thrilled to have this talk, which is maybe a little different than some of our spatial talks, looking at proteomics and the like, but really thinking about how mutations influence single cells and their disposition within tissues. So with that, I will just turn it over to you, and we look forward to your talk. You all know the drill in terms of question and answer. I think we don't have a Q&A box, but if any time along the way, just type a question into the chat and we'll get to them either during or after. Take it away.
Thank you so much for such a kind introduction, and it is my absolute pleasure to present some of our recent data to you today. And I do apologize for the technical problem happened last week, and I'm really glad to see some of you still attending the talk today. So I'm gonna put on my presentation mode. I hope you can see my slides okay. As Thomas kindly mentioned, I will talk about some of our recent work on mutational landscape in human somatic and germline cells. So I will start with a very brief introduction, as I know most of you are familiar with aging and consequences on our genome. And this is an area we are actually really interested to study. And from fertilization onwards, obviously every single cells in our body continuously experience damage to their genome. And these can be consequences of multiple different mutational processes, including intrinsic slight defect in DNA replication machinery, or endogenous or exogenous exposure to mutagens, enzymatic reactions, and also perhaps a slight decline in DNA repair machinery during lifetime. So all of these provide a lot of essentially accumulate in mutations in every single cells in our body, and studying these mutations obviously are very important, as it will provide very fundamental information about development and about cellular maintenance, mutational processes that are operative in our genome, and also about, for instance, role of selection and how it might shape different cellular population and tissues in our genome, in our body. And also information about this somatic mutation can tell us how these different cellular processes might be altered by or contribute to cancer, aging, and other disease during lifetime. And in fact, we were very interested to study different hallmarks of aging, including genomic instability, telomere attrition, and some other aspect, mitochondrial dysfunction, which we haven't included in the publication in September, but this is a kind of a follow-up work. So there are different hallmarks of sort of aging we want to start embarking on and ask questions about, and how our genome responds to essentially natural process of aging. And knowing this information, as I said, is very important to understand how cancer might process or other diseases process during a lifetime. As you all know, studying somatic mutations is rather challenging, as essentially each tissue in our body is myriad of small clones. And this makes it really, really hard with the standard sequencing approaches to understand more about how the genome changes. So we really need very good and specialized type of method in order to study somatic mutations. Obviously, the best method would be a direct readout from every single cell from our body, and that would be, in fact, a dream come true for a lot of scientists like me. However, as you may also agree with me, still single cell genomic sort of platform is not really yet capable to identify all the somatic mutations in a great precision. And this is mainly because of the whole genome amplification that cause a lot of false positives, and also there are a lot of genome dropouts as a result. Another method that is widely used to study somatic mutations in the field is sequencing small pieces of tissues in a very high depth. Obviously, this method is not scalable due to the cost, sequencing cost, and also it still has a very sort of a limitation with resolution of the clone size that we can study. So cells or clones that are represented in a smaller size, they are usually not captured in these type of studies. Another powerful method is single cell derived colonies that I'm sure you're familiar with, where we take stem cells and essentially culture it to study a single cell derived colonies. This method is very powerful. However, obviously the spatial information is lost, and this is one of the down part of using single cell derived colonies. Another method is duplex sequencing, which been actually out there for 5-6 years. And recently, colleagues also at the Sanger Institute have developed a new method, NanoSeq method, which has a nano base pair error rate. And this makes it a very attractable method to study somatic mutations. Again, a problem with NanoSeq type method is it only captures 30% at most in terms of the breadth of the genome. So we don't have a full coverage of the genome. So we cannot do certain analysis such as essentially phylogenetic analysis and etc. Another method, which is the one we picked for the study I will present in my later slides, is laser capture microscopy, where we capture a histological structure, which we think it is clonal, to study a sort of a somatic mutations in a kind of a small pool cells, which allow us a better identification of mutations. So we use this method, which is published in 2020, to study 3 post-mortem tissues. These are adult donors who did not have any certain clinical history. And across all these 3 individuals, we collected 29 histologically distinct structures. I should mention, across these 29 histological structures, for some of them, we only captured information from 1 individual, And for some tissues, we have more than one in donors to compare the kind of mutational landscape in that given histological structure. So overall, we capture 600 patches that they consist of 200 to 1,000 cells across this histological structure. And in this video, I'm showing you an example of what I mean by a patch. This is a histological section of a testicular tissues, very thin section, about 10 micron thickness. And within the testicular tissues, we have a seminiferous tubules that I will discuss in a bit more details in my later slides. All you need to know here is the darker color sort of cells are stem cells and committed cells within these tubules. And we think these are the clonal units within testicular tissues. So we can actually select the area here around the seminiferous tubules, and I'm hoping you can see from the video, we are trying here to focus the microscopy, and laser goes about this area that we try to select and cut the area around it. It will eject it into a well where we can perform whole genome amplification using our low input DNA pipeline. So we use this sort of system to study these 600 patches that they come under 29 distinct histological structures. And we asked certain questions that would tell us more about genomic instability, which is obviously one of the main hallmark of aging. And one of the main questions was about clonal dynamics. So what is the heterogeneity across different histological structures that we cut and sequence? In some tissues where we cut a clonal patch, for instance, in stomach gland and in clonic crypts, we observe that each patch seems to be very clonal. What does it mean? So this area that I depicted here and highlighted in green, they usually are populated, or they're always populated by one stem cells. And here is an example. So if we have a clonic crypts or a bits of a tissue where we cut a crypt or when we cut a gland, we usually have one stem cells that populate these structures. And this is just a representation of the stem cells in a crypt that reside at the bottom, and usually it populates the rest of the cells within the crypt. Hence, any mutation we observe from this patch usually has a frequency of about 0.5, similar to a heterozygous mutation. And this is what is showed or confirmed with the variant allele fraction analysis. So each of these distribution I'm showing you here refer to one patch we cut from each of these tissues. And as you can see, all these distributions have a median VAF of about 0.5. So we describe these tissues as monoclonal. We also observe some tissues such as prostate, acini, and also seminiferous tubules of testes to be oligoclonal. What it means is they usually are seen to be clonal. So most of the time they have one stem cells that populate that structure or that patch, while in some cases we have a bimodal distribution, meaning that two stem cells usually populated that patch. So there are a combination of one stem cells or two stem cells that populate these patches, hence we call them oligoclonal. And finally, we find some of these tissues, such as esophagus epithelium and liver parenchyma, to be very heterogeneous in terms of the stem cell contribution to each of these patches. And as you can see, the median variant allele fraction is very much shifted towards the left, which suggests from this cartoonic sort of explanation that a lot of stem cells populated these sort of patches. Hence, these structures tend to be polyclonal. And by that, for the first time, we generated a kind of a landscape of clonal dynamics or heterogeneity within these 29 distinct histological structures. And we show that tissues of GI tract in general, as well as liver, bile duct, and seminiferous tubules and glands in prostate exhibit a rather clonal structures, while a majority of the cell types we analyzed here had a polyclonal structure. I should here mention that there is a bit of a caveat, obviously, with laser capture microdissection, especially for tissues or cell types where we are not guided by histological structures. So, for instance, where we have epithelium, sheets of epithelium cells that there is no boundaries around them, it is very likely that we do not identify a clonal unit. So where we cut, it might be actually a border of multiple clones, and hence we get this kind of a polyclonal structures. So orthogonal methods and future work should shed more light about the clonal structures, specifically for some of these polyclonal tissues that we do not have exact histological structures. Another question we had in hand, and it was quite important, is what are the mutational signatures? For those of you who may not familiar with mutational signatures, essentially different mutational processes in our body generate a unique combination of mutation types that are referred to as mutational signatures. And in past few years, large scale study of pan-cancer has generated a large catalog of mutational signatures across cancer genomes. And a majority of the studies have suggested that signature 1 of 5 are ubiquitous signatures that are present across all the cancer genomes have studied to date. And these signatures are referred to as aging signatures. And there are other signatures, for instance, UV signature, signature 7A and B, that are identified, for instance, in skin cancer. And other signatures such as smoking signature, signature 4, that are observed in lung cancer, etc. So we wanted to know what is the mutational signature in normal genome and in precancerous era. And this is what we have extracted from the genome of the normal patches that we have studied. As you can instantly see, we have observed the 2 ubiquitous signatures, signature 1 and 5, to be present across all the tissue types here, similar to what observed also in cancer study. So signature 1 is likely due to deamination of 5-methylcytosine, and signature 5 has unknown actually reasons, but it is a clock-like signature. We do also observe signature 18. It is shown in orange to be quite prevalent across most of the tissue types, cell types we have studied here. So it was present in 19 out of 29 cell types. So we think actually signature 18, which is an oxidative damage signature, to be another ubiquitous signature present in normal cell types. Despite these ubiquitous signatures, we do also observe several tissue-specific signatures. For instance, the red signatures that we observe in some of the clonic crypts and appendiceal crypts are signature of Colibactin, which is a strain of E. coli, and previously has also reported in study of clonic cancer and normal colons. We also observe, for instance, UV signature that are shown in brown and dark green to be present in skin, but we observe it only in skin epidermis and also skin hair follicle. However, we don't find this signature in skin sebaceous gland. This is potentially because sebaceous gland reside at a lower layer, and hence they are more protected against the UV light. Another interesting sort of a signature that brought us a surprise was signature of chemotherapy, signature 35, that we observed in clonic crypts and also appendiceal crypt from one individual, the oldest individual. And we did not have much clinical information about this person. However, going back to clinical history, we realized that he had received chemotherapy only 2 months prior to death. And it was actually quite surprising how we can observe chemo signatures in a very short span of time in his clonic crypts. So, despite the fact that here we observe several ubiquitous and tissue-specific signatures across normal cell types, something which was an interesting sort of a conclusion or results to us was that the contribution of the two aging and ubiquitous signature, i.e. signature 1 and 5, seemed to be partially independently regulated in different cell types. So here we show the proportion of signature 1 and 5 contributed to each of these cell types. And as you can see, in several cell types, such as in clonic crypts, in appendix, or in small bowel crypts, we observe that signature 1 has almost equal contribution to signature 5 in these cell types. However, in other cell types, such as in seminiferous tubules of testes, there is a greater contribution of signature 5 and a lot less contribution of signature 1. So although these processes, cellular processes that give rise to these two signatures are identical across all cell types, it seems that their regulation or their contribution independently controlled, and how they are controlled we still don't know, and we hope some of the future experiment can shed more light into this mystery. Another aspect which we did look into it was driver landscape. And I must say most of the cell type that we studied here showed almost none or very small number of driver mutations. And majority of them were hotspot canonical drivers in BRAF or GNAS in appendiceal crypts, for instance, PTPN 11 in seminiferous tubules that were reported previously, FOXA1 in prostate, KRAS in small intestine, and TP53 in esophagus. We also observed some truncating mutations in age recessive cancer genes. Also, we did observe several focal losses and copy number aberration with NOTCHI and TP53 mutations. So I'm not sure if you can see this perfectly, but here in the first plot, we have a missense mutation in NOTCHI, and it has a subclonal 9q loss. Again, we have a TP53 missense mutation that have a 17p loss or copy number aberration. And then we have another missense NOTCHI that has a neutral LOH in chromosome 9. So you don't see much changes in the copy number, but we do observe deletion of the minor allele. And finally, we have both NOTCHI and P53 mutation with a 9q loss. It's a very small section of 9q, almost invisible, about 4.8 megabases. So overall, in terms of the driver landscape, what we observe is almost none or very small. However, I should mention these are all whole genome data. Hence, we don't have a perfect resolution to identify driver mutations, and perhaps deeper targeted studies or exome sequencing would give us a better information about driver landscape in normal tissues. We also asked about essentially structural variations, and here different classes of structural variations are demonstrated in different colors, and each of these gray plots shows how many of the patches we analyze for different structural variation. And we do see variation in terms of DSVs across different tissues between individuals and within individual as well. Interestingly,
就那天一早晨，我是这边变化的哈。
I guess someone's microphone is not muted. So what we see interestingly is in clonic crypts, there is a lot of structural variation across all three individuals, with a lot of retrotransposition event in the 70-year-old male that we studied here, which was quite actually interesting. We also looked into mutational burden and how single nucleotide variation and indel changes between tissues and also within an individual. So here, each individual are shown in different shade of gray. And as you can see, there is quite a lot of variation in terms of the number of single nucleotide variation and indels across tissues and also within individuals. In order to look at the mutation rate, we had to limit our study only to cell types that essentially are clonal. So this is going back to my initial slide, that some of the cell types we analyzed seem to be very polyclonal, and hence they are suboptimal for mutational rate analysis. With that respect, for mutation burden analysis, we only took forward cell types or the patches that specifically had a variant allele frequency of a clonal sort of patch. So these are all centering the red line centered around 0.5, and we use only these patches to infer what are the mutational rate. And as you can see, the rate estimate only limited into very small number of cell types across the 29 histological structures. Nevertheless, what we observe is a huge variation in terms of mutation burden across the tissues and also within an individual. So if you follow, for instance, the darker sort of a color, you can see there is a huge variation in terms of mutation rate across different tissues from one given individual. And interestingly, clonic crypts had a highest mutation rate. So in average, we observed 54 new mutations, specifically SNVs per year accumulating in colon. And on the other spectrum, we had seminiferous tubules of testes with only 2 mutations accumulating per year in this tissue. Another hallmark of sort of aging that we wanted to study was telomere shortening and how it might correlate with the tissues we studied here. And we observe again substantial variation in the absolute telomere length between cell types and individuals. And there was an, in average, about 76 base pair loss of telomere per year across somatic cell types. However, testicular tissues, specifically seminiferous tubules in testes, seem to have a higher telomere length. And when we compare it with other somatic cells, actually within same individuals, we did observe in average 2 kb longer telomere length in the seminiferous tubules compared to their somatic counterparts. And this observation is, of course, is in line with telomerase activity in male germ cells, acting to maintain telomere length over time. But given how variable or how variable testicular tissues was with other sort of somatic cell types, specifically clonic crypts, we decided to have a deeper study of variation between spermatogonia stem cells and clonic crypts stem cells specifically. And for this, we recruited another 11 individuals where we had taken their testicular biopsies, as well as 5 of these individuals, we also had clonic crypts biopsies. And we generated another 300 whole genome data from these clonal patches or clonal units. And we compare the mutation accumulation in these 2 tissues. So here on the x-axis, we have the age of each of these donors. So each data point here refer to average mutation burden in each of these individuals we have studied. So we average all the patches per individual into one data point. And I want to draw your attention in the unit on the y-axis. In testes, we have mutation accumulating in order of hundreds, while in colon, we have mutation that accumulate in order of thousands. And in fact, when we compare the matching tissues from the same individuals, in average, we do see at least 27-fold lower mutation burden in testes versus colon. And this was actually quite interesting for us, such a low mutation burden in seminiferous tubules of testes. And just to give you a brief explanation about the structure of this tissue, testicular tissues comprises of these long tubules that consist of stem cells, spermatogonia stem cells at the periphery, and we have committed cells that generate spermatids towards these tubules. And there is a massive scale of daily sperm production, which put a high demands on testes and also committed cells within this hierarchy of stem cell structures. But despite this high demand, actually, it is very interesting that the mutation rate is still very, very low. And previous studies have shown that some of the mutations in stem cells of testes, those spermatogonia stem cells, can confer a selective advantage to that stem cells, which brings a proliferative advantage, so they can proliferate faster and essentially take over the entire tubule sometimes. And in some cases, they can actually cause germ cell tumors or tumor in an individual. And in other cases, they can be carried by the sperm and predispose children to congenital or skeletal disorders. These are some of the genes have previously published, such as FGFR2, FGFR3, and RAS, that they can cause Apert syndrome, achondroplasia, and Castello syndrome in children. So we were really actually interested to know how, with the information that essentially somatic mutation can confer clonal advantage in this tissue, how the mutation rate actually compared with germline de novo mutation rate. So germline mutation rate is usually studied or inferred by pedigree study other than evolutionary study. And this is something that actually we exercised a few years ago, where we sequenced multiple children from the same family without any clinical history. And we compared the blood genome of the blood of these children with their mom and dad to identify new mutations that essentially must have happened in the sperm and egg of mom and dad. And these are the three families we studied in the past. And back in 2016, we identified that in average, individuals are born with 75 new mutations, and 2/3 of these new mutations are assigned to paternal germline or the sperm of the father. And this is potentially because of the continuous spermatogenesis in male germline. And what was interesting back then is when we compared the mutation accumulation in the germline of each of these that are shown with different color, we observe also variation, a slight variation in the kind of the rate of mutation accumulation in these men. But what is important is we do see, or we did report, an average of about 2 new mutations that usually accumulate in spermatogonia stem cells or in sperm of men. And this is what we observe from pedigree study. So we wanted to compare our study of inferences from the testes and seminiferous tubules with the Pedigree study. And in fact, we used data from DCODE, which is a larger trio study, about 3,000 trios, if I'm not mistaken, and we regressed out the maternal contribution. So here, the purple data points I'm showing you is only the paternal contribution of male germline de novo mutations. And here in the green, we have the haploid mutation rate being inferred from studying of seminiferous tubules. And something which was really interesting, our mutation model comparison between the two data sets suggests that the rate of mutation accumulation is very similar. So pedigree data suggests about 1.3 new mutations per year to be accumulated in sperm or spermatogonia stem cells. And what we observe is very much in line, 1.3 mutations per year. The reason I found this actually very interesting is we knew that germline mutation rate has to be kept low because of its evolutionary sort of role, and it is obviously very important to have a very good genome integrity for the kind of next generation. But one idea we had about the low germline mutation is that this low germline mutation maintained at the time of conception. So a very strong selection on sperm samples that had a lower mutation rate to proceed with the conception and development. However, what actually this study suggests is the low germline sort of mutation is a phenomenon or feature of entire stem cells in the testicular tissues, and the low germline mutation does not seem to be a result of selection of the sperm with lowest mutation rate. So there must be other factors that maintain low germline mutation rate in principle. So this was actually quite an interesting sort of observation for us. And we wanted to follow this more by exploring the existence of DNA damage or repair mechanism that may vary between normal cell types that we studied here, and also whether testes have a different sort of a variation or maintenance compared to other somatic cell types. Also, as a proxy of germline, we did include 13K trios from Genomic England study, as well as a rare variants from NOMAD as a sort of a proxy for germline mutation. And first, we asked about the strand bias, whether we do observe accumulation of mutation in certain strand in the transcribing strand compared to non-transcribing strand. And this was this study, or this sort of correlation analysis was motivated by a study a few years ago now, where they suggested transcription couple repair pathway is a reason for, can be one of the reasons for maintaining low germline mutation rate. What we observed to our surprise is that actually transcription, there is not much of a variation in terms of mutation burden on transcribing and non-transcribing strand in the testes, germline proxies, and compared to other cell type, somatic cell type counterparts. And in fact, we do see quite a lot of biases in esophagus, and this is the reason is actually one of the individuals seemed to be a heavy drinker, and I'm happy to discuss it later on if someone is interested. We also, so the conclusion from this part is essentially transcription couple repair is not exclusive to the germline, and it cannot be the entire reason for maintaining the low germline mutation rate. We also then ask about genomic region, genic region, whether we observe any variation in germline in mutation accumulation in different regions of the genome, for instance, in exonic versus intronic and intergenic. And in all the somatic cell types we studied, we observed lower mutation rate in exonic region. And this is similar to what been shown in cancer study. And it does make sense. Obviously, exonic regions of the genome is very costly to cell types, somatic cell types, and needs to be protected. Interestingly, we don't see such observation in seminiferous tubules of testes, neither in pedigree data, and it seems that entirety of the intergenic, intronic, and exonic regions to be protected. We do see a slight depletion of mutation in exonic region in the nomad data, and this is because of the other evolutionary forces, for instance, gene conversion. So I'm not gonna go through too much details, but this observation was expected. We also ask about the expression, whether genes that express at a higher level accumulate lower or more mutations. And in fact, we do observe an inverse correlation with mutation accumulation as gene express higher. So in highly expressed genes, we do observe less mutations in somatic cell types. And we got this expression profile actually from GTEx, based on the cell types that were available. So for these two cell types, we could not identify expression profile to perform this analysis. However, in germline proxies as well as in testes, we did not observe any correlation. So essentially the low germline maintenance seemed to apply for all gene sort of expression profiles. Finally, we checked a replication timing, and we did observe a positive correlation with mutation burden and late replicating regions. So they are shown in darker color in somatic cell types. And this can be due to the fact that in late replicating regions, there is not enough time for repair machinery to act, and hence we do observe a higher mutation burden and accumulation. However, we don't observe such a strong correlation in testes, neither in germline proxies. So I think overall, what this sort of correlation analysis tells us, whatever cellular operation that maintain low germline mutation seem to be very much different to other somatic cell types and counterparts. And in some extent, every single region of the genome in germ cells to be very important and tightly protected, or they may have actually a superior repair mechanism to essentially making sure that mutation rate remains really low in this tissue. So I'm running out of time, and I'm just going to quickly summarize my talk by few points. Essentially, from this study, we did observe, or we show that there is an extensive variation in both clonal dynamics as well as mutational burden, both across individuals and within an individual. However, new studies, especially deeper study with a larger number of cell types and autonomical methods, should expand more on these variations. This is an ongoing work across the CHASM department and colleagues at the Sanger Institute. We do observe low germline mutation rate to be an intrinsic feature of the male germline compared to the soma. And there can be factors such as high fidelity DNA repair mechanism or some other sort of a protection that essentially maintain this low germline mutation. Again, there are some ongoing work in our group to explore this area and identify a bit more sort of a mechanistic sort of processes that may maintain a low germline mutation rate. And finally, we do observe that ubiquitin signature, specifically signature 1 and 5, to be independently regulated across different cell types. Again, I'm hoping that in future we can show what are these regulatory factors and their importance in cellular maintenance. And by this, I will conclude my talk, and I would like to acknowledge my colleagues at the Sanger Institute, my team, who essentially contributed to this work, all our external collaborators and funders of this study, and by all means, thanks to all the donors and their families for actually making this study happen. And thank you all so much for your attention, and I'm happy to take any comments or questions that you may have.
Wonderful. Thank you very much, Raheleh. I will open it up to questions and let folks chime in. And if no one's going to jump in immediately while you're all thinking of additional questions, and again, you can feel free to type them in. We'll read them out as well. I have lots of questions. I'm actually going to talk to you immediately after this, so I don't want to take all the time. But let me maybe hit the last section you had first. Is germline effect is a very interesting effect, and I know we don't know all the answers, but you seem to focus a lot toward a molecular solution as opposed to what I might describe as a cellular solution in terms of. It could be that they're more efficient in these germline cells at repairing, but it could also be that they're more susceptible to mutation accumulation and cells are. So what's your thought about a tissue cellular versus a molecular mechanism?
It's a very actually good point. Maybe my discussion was heavily centered around molecular mechanism, but I do agree at this stage, we don't have enough information to expand on the cellular sort of aspect that might contribute to low mutation burden. It might be actually some of these sperm or spermatogonia stem cells that accumulate a lot of mutations are not tolerated and being lost or do not contribute to sperm pool. So we are actually trying to look into this space by studying some sort of a phenotypic type of stem cells in spermatogonial or in testicular tissues that might lead a little bit more sort of information on that point. It's a very good actually suggestion.
And in terms of tissue dynamics, is it understood, and I apologize, this is a fairly naive question, but is it conceivable or known how many stem cell divisions those testicular cells may have gone through? Are they protected against going through more divisions as compared to other tissues?
So all the information we have currently about the cell divisions are very much bound to sort of a staining and observing the stem cells. Usually majority are based in mice. There is 1 or 2 studies suggested they work on human stem cells, and there is a wide sort of spectrum of cell division estimates for spermatogonia stem cells, something as frequent as every 16 days, all the way to once or twice of division per year. So there is a large kind of a variation. However, we did compare sort of a mutation rate with different cell division sort of estimates between colon and testes. And what we see is even if the cells do not divide so much, maybe once or twice a year at max, still it does not explain fully this sort of a lower mutation rate. So that's why we think there are other sort of points that help with the lower mutation rate other than cell division.
Great. There's Pat O'Brien has posted a question in the chat, and you can read it along with me if you'd like. Can you, very interesting talk, Raheleh, can you provide any insight into female germline effects with regards to de novo mutation? And what can you tell us about mitochondrial mutations and or oocyte mutations?
Hi, Patrick. Thanks for the very interesting question. I guess I did not deliberately mention about female germline because the female germline mutation are quite lower compared to the male germline and makes it a lot harder to study. We have some work in the pipeline. From the obviously trio study, we have shown, or specifically a few studies from Matt's group specifically focused on female germline. So we do observe sort of a age correlation with maternal germline. The effect is a lot lower compared to the male germline. We do observe about 0.5 new mutations per year. In terms of studying oocytes and somatic mutations, as what we did in sperm, is a little bit harder, needs other techniques to study it, and currently this is a work in progress. As for mitochondrial mutation, this is very interesting. I did mention about it, but I did not actually refer to it in my talk. We have currently done some mitochondrial analysis across different tissues, and what we see is mutational landscape and spectra to be very comparable between the germline mitochondria as well as somatic cell types. However, these are very early data. We do observe some variation in terms of mitochondrial copy number with age in the germline that we do not see with the somatic cell type. So as one age, we do see more mitochondria for some reason, at least in seminiferous tubules, and we do not see such a correlation in somatic cell types.
Other questions? Either type it in or feel free to unmute yourself. So June Lee has asked, what do you think of Italiani's report of lower germline mutation rates for genes highly expressed in male germline?
Yeah, I really like that paper, and that was the inspiration for the first sort of part of the correlation panel in terms of mutation burden with correlation with transcribed and untranscribed region. However, we failed to replicate their observation, and there was another study actually prior to us where they identified couple of sort of analysis they did not consider, which might have actually suggested a different sort of observation. So when we corrected for some of the variation we observed in terms of gene expression, we were not able to replicate this observation. So we do see actually gene expression couple repair pathway exists in the germline, but we don't think this is exclusively working the germline, and it's very comparable to other cell types. So it cannot be the only explanation for lower germline maintenance.
Great. We still have a couple minutes left, so I want to ask you kind of a strange question, but it gets back to the fact that this is our single cell spatial analysis program. And one of the things that in all your discussion of clonality, and some patches are clonal and some patches aren't, and that there's this lineage issue that's going on in tissues. It just, and then we've got all the spatial techniques that are measuring things. I'm curious if you know of or have speculations about ways that that lineage pattern could influence people's results that they're getting out of these spatial transcriptomic or proteomic approaches.
It is really, I think, kind of a hard to address. I guess best method would be a single cell, and in fact I've been doing some single cell analysis on different aspects, and I really love, and I hope in the future kind of the community pick up with a kind of a method that we have a precision to capture single cell level sort of mutation. I think phylogeny driven type of analysis would definitely help us out here. However, my knowledge is potentially very limited in that aspect. We did try some sort of a phylogeny construction using single cell data we generated a while back on the brain, and the resolution is really, really low. This is mainly because of a lot of dropouts that we had across different cells. We were not able to generate a complete phylogeny, but certainly that's a way to go. It definitely helped us to actually remove the spurious type of mutations and maybe drive a kind of a more constructive sort of a conclusion and analysis. But yeah, I found it very difficult actually. Maybe having a sort of a multi-omic single cell sort of approach help us out here with a sort of a guided by phylogeny to remove some of the excess false positive. That's another possibility that maybe we can explore in the future.
Other questions from people? If not, we're getting close to the hour, and I think we'll just thank you again for a really nice talk and some very interesting work, and hopefully some different perspectives for a few of the people on the seminar. And we look forward to hearing more in the future. Thanks very much.
Thank you for having me.